Assalamu alaikum wa rahmatullahi wa barakatuh. Dear all, this is the first lecture and the last lecture in about mycology or fungal infection, which we also call mycosis. So was meant to be an introductory lecture, but also because this is the only lecture we have about fungal infections. Unfortunately, we don't have this time and space to accommodate more than one lecture in mycology in this course, but please bear in mind that fungal infections are relatively common. Although we don't see them in tertiary care hospitals, we do a lot of systemic infections we do see in immunocompromised patients, but we tend to see the superficial type of infections quite common in the primary health care. And the reason is that fungal infection is not very severe because the immune system has learned to fight against this kind of infections over time. So that's why the severity of these infections are not as uh, we see in other other microbes like bacteria, viruses, and also parasites. So in this lecture, we are going to go through a lot of different topics because we need to cover the growth conditions and reproduction of fungi. We need to discuss the categories of fungal infections. We also want to differentiate cell wall structures of those from bacteria, what differences they have from bacteria. And we want to list the common mycosis or clinical manifestations of fungal infections. And then we want to describe how we diagnose and treat different types of fungal infections. Treatment-wise, we are not going to go through the details since we're going to have a separate lecture on the antifungal treatment. That fungi actually are found in the environment and they are almost everywhere. They are even common in the environment more than in our bodies. These are a whole branch of microbiology, but we are going to only have this lecture. So please try to go through these slides very carefully and try to get the most out of them. Fungi are aerobic, so they need oxygen to survive. They are facultative anaerobic sometimes, but they are never strict anaerobic, so they cannot survive if there is no oxygen. And most of them actually live very freely in the soil, as I said, sometimes in water, in air, for one exception, which is Candida albicans. As I mentioned also before, that this is part of the human normal. Most of the fungi are actually saprophytes. So saprophytes means that they actually need to decompose the organic material that they live on in order to obtain the basic molecular structures to be able to survive. There are many uses for these fungi in industry as well as in medicine. So in general, we can use fungi to ferment bread, wine, or soya sauce. So it's called fermentation, which is the conversion of sugars in different types of products or fruits, for example, into alcohol. And then from alcohol, it converts to acids. So that's fermentation. Secondly, we can use it in medicine for production of antibiotics. Like, penicillin is one of the common antibiotics that are obtained from Penicillium fungi. And there is also medicinal mushrooms, which is one type of mushroom that is can be also consumed as medicines or as regular food. However, the presence of fungi sometimes can be harmful because it can damage or spoil the foods and fabrics if they are moist, and they can cause major plant diseases like white patch on the leaves of the plants that you see sometimes. And most importantly, that what we are going to concern in this lecture is it's actually cause some human diseases or what we call mycosis or fungal infections. So where can we find these common fungal growth is actually you can see them in humid or wet places like toilets, for example, at home. In humans, but we can also can cause superficial infections of the toenails, of the fingernails, and also the most important and most common type of fungal infection, which is the hair dandruff. So actually, fungi can exist in two different forms. They can exist as yeast or mold. So molds, for example, as you can see here from this picture here, means that they are multicellular, uh, larger growing colonies. They have long filaments like hyphae, and sometimes they can form mass of hyphae that we know as mycelium. On the other hand, there is what we call dimorphic fungi, which are like histoplasma, can exist in two different forms, like yeast at 37 degrees, which means that when it exists inside our bodies, it can multiply in the yeast form. But once it's outside, like at room temperature at 25 degrees, it can exist as molds. This dimorphic feature does not happen for all molds, but some of them. How does these fungi reproduce? The major reproduction mechanism is by spores. So most of the medically important fungi, they propagate or reproduce by asexual spores known as conidia. And these asexual spores actually form at the sides ends of specialized structures. As you can see in the picture here, they can be in very different shapes and also in very different identification structures. It's the very, very important feature for each one of them for us as microbiologists, because these specific identification structures are very important for diagnosis of fungi. Each fungus has a very unique structure that we can observe under the microscope. So asexual spores can exist in different shapes. And from A to F here, I have listed different shapes, as you can see from the picture. A is blastoconidia and pseudohyphae. B is chlamydiospores. C is arthrospores. D is sporangia. E is microconidia, and in F we have micro as well as macroconidia. And I've explained before in the practical that micro means small conidia, macro means the larger conidia. For the cell wall of the fungi, it's very important to note that there are some structural differences between the cell wall of fungi and the cell wall of bacteria. So fungi is the plural of fungus. So fungus is one and fungi is many. So fungi are not, unlike bacteria, they are eukaryotes. So this means that they have a little bit of different structures. Like in this case, they have a very well structured nucleus. So there is a DNA and RNA that is enclosed with a nuclear membrane. There's also ribosomes in the cytoplasm that are similar to the human ribosomes in size. And the fungal cell membrane has some unique structures like ergosterols, and the fungal cell wall also is unique. So the ergosterol here is different than the ergosterol or the type of sterols in the human, which are cholesterol. So these ergosterols, as you can see in the top picture here, these are phospholipids that are important for three main functions. The first one is to protect the cytoplasm. Second one is to regulate the intake and the secretion of the nutrients, and also to facilitate capsule as well as cell wall synthesis. So they produce the precursors or the building blocks for the cell wall. There are two important cell structures for us to know. Why? Because we use these structures for the diagnosis of fungi. The first structure is chitin, which is the fungal cell wall, and it's not a peptidoglycan layer. It's different. It's a polysaccharide layer that is having long chains of an acetylglucosamine. And there is another structure which is important in the polysaccharide as well, is known as the beta glucan. This is a long polymer of D-glucose. This beta glucan is very important because, again, it is a site of action for certain antifungal drugs like caspofungin. So again here, penicillins will not work because they need a peptidoglycan which does not exist in fungi. I put this table for you to be able to study it and understand the major differences in the structures between fungi and bacteria. Please go through all of these uh, parts and make a distinction between the two. So it's very important for you to understand these major differences. Now we move on to this very basic morphology element of fungi, which is, I put it here for you to understand what do we mean by threads or hyphae, like you can see here in the first picture here, A. And we have septate or non-septate, which means they are separated by the cytoplasm or not. If there is separation, we call it septate. If there's no separation, we call it non-septate. And if they form tangled threads just together in a collection, we call them mycelium. So mycelium is basically a web of branched hyphae. In the third C picture here, we see the budding yeast. And in D, we see pseudomycelium, which means that they are similar to mycelium, but it's not a real mycelium. Now move on to the classification of fungal infection. This part is very important because it tells us that we can classify it medically based on the site of the infection into very major 4 sites. So medical mycosis, we divide it into cutaneous, subcutaneous, systemic, and opportunistic. And we're going to go through each one of them with giving examples. Please note that when we give examples, this means that they are not the only infection, they are just examples of that group of infection. So the first one is cutaneous mycosis or dermatomycosis or dermatophytosis, also known as tinea or ringworm. So this does not mean it's a worm infection, but it forms a ringworm or a ring structure, and that's why we call it ringworm. This type of infection is a chronic infection, which means that it stays on the skin for a very long time, and it occurs in very warm, humid areas of the body. And this actually happens in different parts of the body. So each part will have a different name based on the type or the site of the infection. So ringworm lesions, they usually are circular and they contain papules, which means that they have very small raised lesions on the skin with vesicles sometimes, and the surrounding skin is very normal. So you can see them very clearly. To understand what I meant when I said different site of infection has different names. So for terminology, if I say tinea, tinea is the first part of the word, which means that it's a fungal infection. The second part, it describes the part of the body. So corporis means in the trunk of the body. So tinea corporis occurring on the trunk, like chest or abdomen. Tinea capitis means that it happens on the scalp or the hair. Tinea cruris in the perineum. Tinea pedis in the foot, and that's why also the other name is called athlete foot. Tinea euginum, which is in the nails. Tinea barbae, which is on the bread and on the beard and mustache. So you can see here on this picture there are red scaly rashes, and there is central clearing, and the area surrounding it is normal. So let me give you here one example of a case. This is a 40-year-old dark-skinned male who presented with 1-month history of skin discoloration. This is actually an infection. If dark skin, what you see here, there is a hypopigmented area, and this is known as tinea versicolor because it changes the color of the skin. On the top picture, actually it's the opposite. If the person has a very light skin, colored skin, the lesions will be darker, so hyperpigmented. So it can be either hypo or hyperpigmented. This type of infection is caused by a fungi called Malassezia furfur. It's very common, and we see this as hypopigmented areas on the chest, on the neck, sometimes also on the face. This happens very frequently in hot, humid weather. It's very common also here in Oman, and we see a lot of cases in primary health care. And this type of infection usually happens during summer because of the hot weather, and it's likely to occur between siblings because of the sharing of the devices. We treat this type of infection with a topical ointment called myconazole. Now we move on to the another type of tinea infection, which is called tinea nigra. Can you see this picture here? It's actually showing you that there are dark areas that are infected with this type of fungus. It's on the keratin layer of the skin. It gives a very brown color, and this is caused by melanin pigmentation in the hyphae themselves, that they will give this dark color. The causative organism is Cladosporium wernicke, which is actually acquired from the soil. So if the person has injured skin and they touch or work on the soil, like farmers, for example, this is likely to be transmitted to the person. So the previous examples that I gave you all are cutaneous mycosis. Now we move on to the second type, which is subcutaneous mycosis. They are very similar to the cutaneous ones, but this actually is likely to occur in the deeper layer of the skin. It's very common in gardeners to have this type of infection because they work and manipulate with soil, and they are very likely to get injuries. They are caused by fungi that vegetate in the soil, and after trauma, these spores from the soil get access into the skin and then penetrate to the deeper layer. So when these spores penetrate the skin, they actually form what we call pustules or ulcers, and then they can escape to the draining lymphatics, and that's why what we see here on the picture, you can see a line of nodules occurring at the same line of the lymphatic system. So this is what we call sporotrichosis. Now we move on to the third type of fungi, which is the systemic mycosis. So what we mean by systems, that it affects the internal organs. And actually, these infections can happen through the inhalation of the spores, so from the respiratory tract, of any dimorphic fungi. So if they are existing in the mold form in the soil, they can gain entry to the respiratory system, and then they can, inside the body, exist in as yeast because they are dimorphic. But most of these lung infections are asymptomatic and self-limiting. So this means that the patient might have very mild symptoms. He will not come to the hospital unless there is severe manifestation. A very good example of this is pulmonary histoplasmosis, caused by a histoplasma type of fungi. Now we move on to the fourth type, which is opportunistic mycosis. So this opportunistic mycosis actually similar to what we have explained in bacteriology. Opportunistic means that this type of infection happens only in immunocompromised host. Otherwise, in normal individual with good immunity, it will not happen. So this example for this is cancer patients who are on chemotherapy, or patients after organ transplant, patient undergone surgery, or AIDS patient. Examples for this, like in AIDS patient, we commonly see Cryptococcus neoformans, and this type of yeast I have mentioned and also explained in the practical slides. Candida species is another opportunistic mycosis that happens in reduced immune patients. Aspergillus species, as well as different types of, histoplasmosis can be also one common opportunistic mycosis. I have also mentioned a similar case in practical slides, but I'm going to give you this example again. It's a 50-year-old woman who receives chemotherapy by a subclavian catheter, which means in the central line. She was receiving treatment for acute leukemia, and then she had a sudden onset of blindness in her right eye. The total white blood count was high, and blood culture showed budding yeast. And these budding yeast showed germ tubes. So in the practical slides, again, I have described to you the germ tubes. So the diagnosis for this lady was actually infection inside the eye caused by a Candida albicans. So how did this infection happen? It's actually because the patient has a catheter inserted. So from the catheter, this infection actually spread through the catheter to the blood, and then it has reached the eye. And because this patient has very low immunity because of the chemotherapy, the immune system was not able to fight for this infection. It's actually caused Candida albicans to become active. We have also mentioned before that Candida albicans is part of the normal flora of the skin, and it actually gains access to the catheter if there is break on the skin. So basically, germ tube test is a very important test for diagnosis of yeast. You incubate the serum of the patient for 3 hours at 37 degrees, and what you observe is the germination. So this is the mother cell here, and there is a germ tube that is protruding from the mother cell. And these are hyphae actually, and this is diagnostic for yeast. So on gram stain, Candida albicans can give you a gram-positive reaction. However, you will know that this is not a bacteria because of the size. They are relatively larger than the bacteria. You can also see pseudohyphae. Pseudohyphae are chains of elongated budding yeast, which means that they are not real hyphae, but they are yeast that budding cells that have become very long. Other types of manifestations of mucosal candida is oral thrush, which happens on the mucosal surface of the oral cavity. And this is likely to happen in patients with predisposing factors like obese patients, patients with diabetes mellitus, or patients who are using steroids or antimicrobial therapy, which means that some antimicrobials can be suppressing the growth of the bacteria. This allows the overgrowth of the candida. Also in immunocompromised patients on chemotherapy become also immune suppressed. And because it happens in the mucosal surfaces, it can also happen on the mucosal surfaces of the vagina, and it's very common to have vaginal candidiasis in pregnant ladies. Cutaneous candida is also known as candida intertrigo. So intertrigo means that it can happen in the axilla, inguinal regions, and sometimes between the webs of the toes and fingers. They form what we call as erythematous plaques, and they have very small erosions that are very painful. So how do we actually grow a candida? We have also mentioned this previously, but just to remind you that we use saborodextrose agar. This is the usual or the simple media that we use to grow most of the fungi. Yeast actually can give you very distinctive creamy to white, flat or dome-shaped colonies. They are sometimes dry, sometimes they are waxy surfaces. Now, just to give you one more example of a severe type of mold infection, we have a case study here of a 56-year-old male with leukemia who was in chemotherapy, who was neutropenic, which means he had very low white blood cells, and which means he's very immunocompromised. He developed fever, cough, difficulty in breathing. This is the chest x-ray, CT, as well as the histopathology of the patient. As you can see here, there is a nodule on the lungs of the patient. So the sputum culture of this patient actually grow. What we see here is a type of mold we call aspergillus, and this type of infection is called aspergillosis. So aspergillosis is caused by aspergillus. Different types of species can be seen, but Aspergillus fumigatus is the most severe type of Aspergillus, and it is most frequently involved in the lungs and also paranasal sinuses. It can cause spread from the lungs to the brain, kidneys, liver, heart, and also bones. However, this does not happen if the patient has good immunity. It only happens in immunocompromised hosts, like if they have very low neutrophils or they have defect in the white cell count or neutrophils if they are receiving chemotherapy or systemic corticosteroids, like in the case of patients with autoimmune diseases. So that's it for the common fungal infections. Now, before ending this lecture, I just wanted to highlight a few things. So, are these fungal diseases communicable? Do they transmit from one person to another? Actually, most of the fungal diseases are not communicable. There are, however, some exceptions. So, we have mentioned the infections happening on the skin, like dermatophyte infections or ringworms, the tinea. This can be transmitted from one person to the other by sharing towels and fomites. Candidiasis can also spread from one person to the other. Again, this depends on the hand hygiene, and if you are a healthcare worker like you are nurses, you have to be cautious because hand hygiene again becomes the most and foremost important thing to actually prevent the spread of such infections. And then vaginal candidiasis. Sometimes, if this is like if women has vaginal candidiasis, even if they have transmitted this from their partners, they are usually asymptomatic. So the exact reservoir is not known, but you know, human to human is very rare transmission, but it can also happen by droplets in the aerosols. How do we diagnose fungal infections? The most important one is direct microscopic examination. And what we do here is that we take scrapings of skin or nails and we treat it with 10% potassium hydroxide and put it on a glass slide. And then we can see the septate hyphae under the microscope. If we see this, we can culture it also on sabrodextrose agar at room temperature. Sometimes also in the incubator, and then we take it out of the incubator and leave it in the room temperature for some time. In some cases, we can also use what we know as fluorescence stain. So fluorescence actually, like the picture I showed you in the slides of the practical, this can actually, when you subject the infected site to ultraviolet light or Woods lamp, it can highlight the infected area very well. Another test we can use is the DNA probe test for certain types of fungi. Serology tests can also be done in some case of antibody detection in some fungal infections. Now, how do we treat fungal infections? Again, fungal infections are treated with different classes of antifungals, but we are not going to go through them in this lecture. Sometimes, if there's large lesions and it can cause deformities of the skin, you can perform surgery to remove that lesion. Or sometimes, if they are causing obstruction in the lungs, for example, or the brain, you can also remove them surgically. The second important thing is the modification of risk factors. And by that, we mean that we have to find out where the source of the infection is coming from. Like some people having aspergillosis, then we have to check maybe they have mold, and cleaning the environment becomes very important. This is not very common here, but in Western countries where the environment is very humid and moist, the mold and mildew is very common on the walls and the roofs, so they have to clean it very regularly in order to prevent occurrence of fungal infections. That Thank you very much for listening to this lecture. I do want to remind you that these are the only introductory slides we have. However, we are going to have the practical session, which is going to cover a few cases of fungal infections. So please go through the slides and prepare your questions. So any things that you don't understand, we're going to discuss in more details in the practical session. And thank you again for listening.